Alright, here we're going to move our way into movement across the cells. We're going to really start taking a look at the structure of a cell membrane. Now, some of the different SWABATs that I want you to be able to take out of this today, the biggest structures today, is identifying the different movements within a cell, the different movements from high to low, materials traveling back and forth, whether that be as simple diffusion, where movements of any high to low solute goes through, facilitated diffusion, the facilitator looking at how that protein that was just embedded in the cell membrane allows materials to pass through. That active transport is also the use of those same proteins, but instead of it going with the concentration gradient from high to low, it's going to go from low to high with the use of the energy in the form of ATP. And then last of those types is osmosis. Osmosis is the only one we're going to talk about with the use of water. That is the diffusion of water. Describe different types of solutions, hyper, hypo, and iso. You're going to also see this in terms of osmotic pressure. So instead of it being the solution of solutes, which is in regards to the tonic, it's a hypertonic, hypotonic, isotonic. We're also going to talk about hyperosmotic, hypoosmotic, and isoosmotic. The structure of the fluid mosaic model. What does it actually look like? How does it become to be? So be able to tell me what the structures are that entail it. So the polar heads, the nonpolar tails, the embedded membranes. Movements of materials in and out. How I can pass a solid, undergo phagocytosis, enter a liquid penocytosis throughout the cell. Explain how proteins play a vital role in the transport, so like above, to facilitate an active transport. And describe how cell membrane interacts with other cells, how they're able to communicate with other cells. So if I'm looking here, listing at the different methods of movement into another cell, this is another one. Explain the process of waves as they move through. Describe the fluid mosaic model, what components are associated with it. Explain the differences between those different osmotic solutions, as well as then identify the purpose of the cell membrane and how it regulates. So it's a structure and function based off of. So in terms of the second law of thermodynamics, what it really kind of covers is it says that in universe we are kind of universe tends towards disorder. So if there's a lot of things on one side, they want us to have separation. So we understand that process that's going on where I have a high concentration over on this left side is wanting to go over to where there's the right side. But in that same sense, if I'm looking at it, so that same sense, notice that there is a lot of water here. There's a lot of water on this side. That water is also going to be moving in the opposite directions. So it can be said that not only is diffusion happening, but so is osmosis. Because if we look at our solution of water that had a semi-permeable membrane, I've got high concentration of sugar on one side. These are all sugar molecules are going to want to go this way to where there's low sugar. So that process is known as diffusion. Now the opposite is said as I have high H2O on this side where there's more pure water. It's going to want to go this way to where there's low H2O. So if this way is diffusion, this way is osmosis. So true and false, diffusion and osmosis happen the same way but opposite directions. That is a true statement. Okay, moving our way into the next structure. Identifying diffusion of two solutes. Each substance diffuses down its own concentration gradient. Now, depending upon how large it is, the particular solute may have an easier chance to get through or not. Now remember, in terms of our structure of a cell membrane, the cell membrane is nonpolar in the tails. So something really big but still nonpolar still can't get through. We need something small and nonpolar is able to pass through with no problem. For example, gases like methane gas or carbon dioxide, carbon monoxide, oxygen. Those types of gases can easily pass through. There's no charges on it whatsoever. But as soon as we start looking at things that have charges to it, those polar structures, they're not going to be able to pass through. And when I say polar, water. Water cannot easily pass through the cell membrane. So it has to have its own pores to be able to do that. So this Brownian movement that we've seen before or we've mentioned is diffusion. So Brownian movement is diffusion, the movement of high concentration to low concentration where no energy is needed. So we always kind of give the example that if you're on top of the hill and you are going to be this person down here and you've got a skateboard or a sled that you're going to go down and you want to go down this big hill, it's much easier to go down than to sled uphill. Okay, sled downhill is no energy. Okay, where you have to go uphill is using a lot of energy. Going back to the cell membrane, cells need inside and out. What it helps to regulate, helps to be able to bring materials in, all our food, sugars, proteins, materials that we need into the cell, and any waste product we can eventually get rid of. Okay, we've talked about the overall structure of the phosphate head. You remember this, if we looked at the basic subunit of it, you had your glycerol portion was your head, and then you had the 3 fatty acid tails. But remember, we got rid of this fatty acid and instead put a phosphate group on it. So that structure there is here. You have the phosphate head and then lipid tails. Those lipid tails are hydrophobic. Head is polar. Okay, hydrophilic, nonpolar. So looking at our particular structure is polarity because we're dealing with charges. Now notice this has a kink in it, so it stays fluid, doesn't stick together because of the double bond. So it would be like an unsaturated fat there, showing on that structure. Semi-permeable membrane means that it needs to control what can go in and what can go out, like sugars, amino acids, lipids, water, salt, ammonia, how permeable it can actually move things through. Now, things that don't have a charge, that can easily pass through, things that are small, but anything that is polar and doesn't have a particular charge, like here, this guy should be able to pass through. It's fairly small, but a large molecule like sugar is actually going to need some pores within it to be able to transport through. So simple diffusion is just something where something really small can easily pass through with no problem. But where you get into water, water being polar needs a protein. So I have to have a channel to get through because water cannot interact down through those tails. It has to work its way through what we call an aquapore. An aquaporin is something that can allow materials to go through. Sugar also uses other pores. We just call this facilitated diffusion. Facilitated diffusion because this protein here is acting as a facilitator, a helper in allowing materials to pass through in and out of the cell, so wherever there's high to wherever there's low. Now, each of these colorations are showing based upon their shape, their particular structure. Now, the yellow is different from the gray, different from the blue, from the pink, from the green. Each of those is different because they each have different structures that allow through. This goes back to the elementary example. The square peg shouldn't fit into the triangle shape. It can't work. Now, each of these have particular shapes that are folded. Now, if we go back to our protein structures, it goes into our primary structure, which is peptide bonds, are the most important. Then our secondary, and then our tertiary structures, and then our quaternary structures. Ultimately, what we are dealing with is the shape, the folding of the proteins that are going to start to take place. And as they start to take place, we get multiple protein sets in it that might make a particular shape, like a triangle shape, that things can actually get through. Now, ultimately, what does it look like? Does it look nice and neat? Or is it kind of blobs all over the place? Now, ever since you were probably a little, we were talking about, it's really going to be this guy. There's a model called the Davison-Bernoulli model that actually looked at it like this, all having these proteins on the bottom and on the top. But that's not going to help us to get materials in through the actual lipid bilayer. Nothing can still get through there. So I have to have proteins that actually can allow the door to travel on through. So it has to be unique form that way. So if I look at this structure of a hydrophilic and hydrophobic protein, we talk about something that's amphipathic. Again, containing both properties of polar and nonpolar. Now if I was to look top down on it, I would maybe have this nice open channel that I could have materials easily pass in through those alpha helices of that transmembrane protein. Transmembrane because it goes all the way across. I've got polar heads down there, polar heads up there, non-polar alpha helices within there, so it can interact in both. So again, it is going to be a amphipathic structure. Okay, so facilitated diffusion allows this protein to be able to pass materials through. So I have a high concentration up here to where there's low. This is the bouncer. There's enough space in the dance club that you want to go to. You don't have to pay the extra money because there's a lot of people outside, little people inside. So we want to get more people into the club. Now, on the reverse side of that is saying in active transport, we still have those proteins that they're going to utilize through, But now there's way too many people at the club. Now there's too many people at the club. So if you go back to the doorman or the bouncer example, I still have a lot of people outside. But there's the inside is even higher amount of people. Well, people still want to get in, so they have to pay. So they use an energy source of ATP to pay, so they can still shove more people into the dance club. So the three different types of movement: passive, facilitated, and active. Passive, no energy; facilitated, no energy but needs a protein; and active, still needs a protein but also requires energy. Low to high, high to low, high to low. Okay, so let's say the same thing there. Okay, what this is saying, gated channels, is just talking about the ion gated. There's a pump called the sodium-potassium pump that is especially important in those that have taken anatomy. Probably remember the polarization, depolarization, repolarization of the cell membrane that allowed sodium and potassium to flip spots to cause an electrical current to take place. So it's sodium, potassium. So when sodium come in, potassium will come out, flip-flopping back and forth, which allowed an electrical signal to be able to pass through. Gated channels that allow things to be materials to go through. In this example, there are actually every 3 sodiums to 2 potassiums. So, in that being said, that we always keep a charge within the structure, and that charge allows for electrical current to be able to pass through. So, for every 3 sodiums, there's 2 potassiums. So here's just an example of that sodium-potassium pump. As you can see over here on this side, sodium would join in those 3 spots, potassium would join here. As sodium would come in, potassium would then leave. It needs ATP, so it needs a lot of energy. So if I'm talking about it in the brain and the nervous system, there's definitely going to be a lot of mitochondria also in that area. Other ways of transport systems, so simple diffusion can pass right on through without any help of a protein. Facilitated needs a protein but still high to low. Active transport where I have a lot inside already but still want to have more, so I have to pay the doorman, pay the bouncer, and I use energy as my currency. Okay. How about large molecules? We're looking at moving large molecules in and out of the cell through vesicles or vacuoles. So this is our phago and pinocytosis. Cell eating is engulfing a solid structure. Cell drinking is engulfing a liquid in structure. So those are 2 different ways that materials are passed through. Both require energy to have that take place. And then exocytosis is another one where instead of bringing it in, I'm actually going to be leaving the cell structure. So here's an illustration that shows phagocytosis eating, pinocytosis liquid, and then cell mediated is signaling, like signal transduction. Okay. Special cases of water. Well, actually at this point we'll stop here. We'll start special cases of water next.